is honestly my first time meeting you.
Yep.
And I'm also a PhD, but my PhD means post-hole digger.
No.
I have a long way to go before they can call me Dr. Matt.
I'll say I can give a little introduction. So I met Dr. Lohman this summer, and very intelligent guy. He's got a lot to bring. And he had this product called Seaweed Max, and I was like, I came back to Matt and I said, God, we gotta use this stuff, and it actually is pretty good stuff. But besides that, Dr. Lohman's very good at what he does, and works for a very reputable company, studied under the great Fred Bilo, and I'll let you take it away, Marcos.
Yep, so thank you Blake. Thank you for the invite to be here, very glad. And I love this format here, it's much more informal, so if you guys have any questions, we can interact much better. I think it's easier than a big crowd, so feel free to interrupt me anytime, okay? As I said, my name is Marcos Lohman. I'm originally actually from Brazil. I came to the US for grad school. I went to U of I, not Iowa State, unfortunately, but I went to U of I, studied under Dr. Fred Bilo, high yield corn and soybean, very focused on soil fertility, plant nutrition. Spent 5 or 6 years at U of I, and after that I started working for Brandt. Was not necessarily planning on staying in the US, but I met my soon-to-be wife, and she's American, and we figured out it was easier for me to stay here than her to go to Brazil, because she doesn't speak any Portuguese. So yeah, that's how I ended up here. With Brandt, I am located at our research farm in Pleasant Plains. So we have a 300-acre research farm in Pleasant Plains. I would like also to invite you guys, if you are interested, to go to our research farm, see what we have there. We have a very nice tour we give with all of our high yield management practices exposed. So it's pretty neat. Blake been there, and I would highly recommend just get in touch with me, and I can arrange that. But anyway, we have this 300-acre research farm in Pleasant Plains. I work at the research farm. I help Eric Winans run the research farm, the trials, where we're going to have everything there. But my work started expanding abroad. Like, I started working a lot in America, but then I went to Brazil, started doing some business with Brazil, trying to bring some technologies they're developing in Brazil to the US and vice versa. And that ended up making me now the International Agronomy Director. So just 1 month ago, I changed my role, and now I oversee all the agronomy done across the world, actually. So our company now sells products in over 80 countries, so we're a pretty big international company, but still 100% family owned, and that's one thing that I really like about Brandt. We're a family owned company. We were born in 1953, so we have over 70 years in the market. We started as a retail. We still have our retail locations in Central Illinois, but we started growing abroad, like out of the retail business into micronutrient, biostimulants, planter box products, adjuvants. So we have a full line of products that Blake works with us as well. So he sells a bunch of the products that we formulate for them. So we're pretty good formulators, and I'll explain a little bit why formulation is so important when you think about micronutrients, when you think about these different products, okay? And please interrupt me anytime if you don't understand or if you don't agree. I don't know everything, but I might be able to help you guys with some ideas and things. Think a little bit outside the box, okay? And we'll be talking about soybeans, high yield soybeans. I really like soybeans. As you guys know, we grow a lot of soybeans in Brazil, so I've been working with soybeans for quite some time. And we're fortunate enough to be working close to Alex Harrell, world record yield holder for soybeans down in Georgia. He utilizes our products, and we have a very good relationship. And we discuss a lot of what he does in his management, and a lot of what he does I'll be talking about here as well. So that really adds up a lot to the conversation. And since we'll be talking about soybeans, we'll be talking about soybean yields. I always like to bring the yield components first, because there's no way of changing soybean yield without changing one of these two yield components. So in the easier way to think about it, yield of soybeans are determined by the multiplication between the number of seeds per acre and the average seed weight. I'll go more in depth and explain how seed weight is determined, but between these two, seeds per acre and weight per seed, which one do you think is more correlated to yield? Differences in seed per acre or weight per seed? What do you guys think? More correlated to yield.
Weight
a lot of people think that, but actually it's not. And this is a study I did during my PhD. We would run over 100 trials a year. So we had a lot of trials at U of I. We had a massive data set. And I gathered a bunch of those data sets together, 1,258 samples from 35 different trials across Illinois from 2014 to 2021. And I correlated the yield component seed number and seed weight to grain yield. And what we found is that seed number is actually much more strongly correlated to yield than seed weight is. And this actually matches very well other literature, published literature there's out there. And there's a reason why. Which one of these two yield components do you think is determined first in the season? Seed number or seed weight? Number. We are managing our soybeans much more to improve seed number than to improve seed weight. And I'll show you guys a graph later on, or an image that shows when these different yield components are being determined. And you'll see we're not really managing past R5. That's when we're determining seed weights. So that's why management practices that we're doing right now in Illinois, and I believe Iowa is pretty similar, are impacting much more grain or seed number than it is seed weight. It doesn't mean that seed weight doesn't change or you can't really improve it. For me, I see it as opportunity. There's a lot of opportunity here. And one very important thing about yield components, and that's true as well for corn, there's compensation between them. So it's not as easy as, hey, let's manage early on, have more seed number being produced, and you get greater yield, because yield components are negatively correlated. I have a typo here, but more seeds per acre usually correlates to less seed weight. And that's the graph that correlates seed number and seed weight. And that's true for corn as well. So if you, for example, on corn, we have three yield components: plants per acre, or ears per acre, number of kernels per plant, and the weight of each individual kernel. If I increase plant population, let's say plant 42,000 plants per acre, usually you end up with less kernels. If I plant lower population, you might end up with more kernels, but you'll end up with lower weight, unless you manage for it. So our goal is to break this negative correlation. Improve first seed number, and keep managing to improve seed weight. And you understand that it's not that complicated to improve seed weight. It is actually complicated in the real world. If you think about it, it's not that complicated, but it is in the real world. How do you improve seed weight? What increases seed weight? Photosynthesis. So we need to be photosynthesizing, we need green leaves to improve seed weight. And a lot of times when we get to the end of the season, we start remobilizing nutrients, we lose photosynthetic capacity, and we lose seed weight. So you'll see that a lot of what we'll talk about later on is managing to improve photosynthesis later on in the season, and that way we can improve seed weights as well. So,
Question on that.
Yes, sir.
Is that correlated to keeping things greener longer?
Absolutely.
Not been my. I'm just curious.
Yeah, because there's no way of doing photosynthesis without being green leaves, and that's what we did a very interesting trial. We do a lot of research on soybean a lot, but we have even more research on corn, and there's a very interesting trial we did with Dr. Dan Quinn from Purdue. He looked at the effect of fungicide and fungicide plus one of our nutritionals on the effect on duration of grain fill and the effect of that on kernel weights. And we saw like impressive results. I can show you guys later, but pretty much slowing remobilization keeps our green leaves green. With that, we do more photosynthesis. With that, we extend our duration of grain fill, and with that, we get more kernel weights. And if you talk to Alex Harrow, he harvested his beans. They were green, not a single leaf had remobilized. He yielded 230 bushels, something like that, per acre. Not a single leaf was yellow. The entire field was green. All the pods were dry. He comes out with, I think, glufosinate or gramoxone, I'm not sure what he utilizes, but he desiccates it and then he harvests it. But he gets to the end of that season, still green, photosynthesizing, and that's how he almost doubled the average seed weight. Do you know what the average seed weight for soybeans is usually in America? Around 150 to 160 milligrams per seed. 150 to 160. Alex Harrow, 274. So almost double. They were massive beans. And on top of that, he not only had massive beans, but he had a lot of beans. The challenge is, how do you make a lot of beans and make all those beans big? That's the biggest challenge, and there's no other way of making them big without green leaf photosynthesizing. And the reason we're not photosynthesizing late in the season will change depending on the crop, but a lot of it has to do with nitrogen fertility later on, with phosphorus fertility later on, because when we need nutrients inside the grain and we don't have the nutrients coming from the soil or through the leaf, where is the plant going to get those nutrients from? From chlorophyll, from the leaf, from the biomass. And then we start remobilizing those nutrients and we lose photosynthetic capacity. Okay, back to the yield components here. So the yield components are negatively correlated, but with proper management, we can break this negative correlation and increase yield. And this is a figure that I wanted to show you guys. And this is one of the reasons why seed weight or seed number is much more correlated to yield than seed weight is. We're determining seed number all the way to R4. Grain fill starts at R5. Who here is managing their beans past R5 or R4? Almost no one. In Illinois, I don't know anyone doing that. We started doing some trials to look at the effect of late season management, and I think that's probably the future, because we've learned how to make big bushy beans, a lot of pods, but then you come to harvest a lot of times and you're like, man, the yield, I thought it was there, it's not. And that's why beans are so hard. And the reason is, you make a lot of beans, but you don't manage to break this negative correlation, you lose seed weight, and then you don't get the yield you want. So I think with drones, I don't think drones are there yet, but we saw so much improvement over the last years. I cannot even imagine in 10 years how drones are going to be. That's going to give us a lot of opportunity for late season management on corn, on soybeans as well. And that's what Alex Harrow is doing. He's going into the field every week applying. He won't spend more than 10 days without going in the field, and he utilizes drones. He'll go with his ground rig until R3. When the canopy closes, he utilizes drones, and he goes out and applies fungicide, biostimulants, and nutritional all season long, keeping that plant healthy and alive to maximize both yield components. So this explains a lot why we see that type of correlation, and I see a lot of opportunity here. So back to these yield components here. So seed per acre and weight per seed is the easiest or the most simple way of looking at these yield components, but we can break seeds per acre down and understand that different variables are actually impacting seeds per acre. So seeds per acre is going to depend on the number of pods per acre times the number of seeds per pod. Seeds per pod usually genetically determined and a lot to do with the environment. We don't necessarily know any management practices we can do or put in place to change seeds per pod. We don't know, oh, add zinc and you increase that. So we consider this a fixed, genetically determined. But the number of pods per acre, this one can really be influenced by management, because pods per acre is going to be influenced by the number of nodes multiplied by the number of pods per nodes. So what I'll be talking about here today are these three yield components. Because seeds per acre depends on the number of nodes and pods per nodes, I'll be talking about how we can improve node number, how we can improve pods per node, and also how we can improve late season weight per seed. So I'll walk you guys through each one of these yield components and how we can positively impact them through management and also some products. And that's not all of it, because we don't have enough time to talk about all of it, but just some things that we are doing to see the yield results that we're seeing. And at the research farm, a lot of these things are working for us. So I was talking to Blake last year. Our average soybean yield is not a big area. We only have 300 acres there, but 100 acres of beans, 200 acres of corn. We have some corn on corn, but 100 acres of bean ended up 102, which is a pretty good yield. Corn, we ended up around 280 last year. Corn yields went down, but bean yields went up, and I think I know the reason why they went up. So let's start talking about node number and how we can influence node number and why node number is so important to maximize soybean yield. The more nodes you have, the more points of attachment for pods you have. With that, you have more pods per acre, with that you have more seeds, and with that you have higher yield. And one of the main management factors that can impact node number is planting date. When you plant earlier, you tend to end with higher node number. A lot of times, because the soybeans are indeterminate, even late planting may get there, but it may get there, those last nodes are not going to put as much pods as an early planted soybean. So these are pictures coming from the University of Illinois, a planting date study, looking at 4 different planting dates and counting nodes. Of course, these are still going to grow, but the closer you plant to the summer solstice, the fastest that crop will turn into the reproductive mode. The moment this crop turns into the reproductive mode, it's diverting energy. So think with me, this first planting crop, until June 21st or 23rd, until the summer solstice, it's spending all its energy growing vegetatively. The moment it starts flowering, it has to divert energy into a reproductive and vegetative. This plant will do that much earlier, so it won't be growing at the same pace because it has to share energy between flowers, pods, and vegetative. So we get an advantage of planting early, and that's why we see an advantage in yield at the end. I think that's also true here for Iowa. Are you guys seeing earlier planted beans being more common?
Historically, yes.
Yeah. For us as well. This is data from the research farm, about 10 years, historical data. It's all normalized to percent of maximum. So all the dots, or the dots and crosses here on the top, 100% means that was the highest yield for that given year. And the rest here is a percentage of that highest yield. We have corn and soybean. You see that soybean prefers earlier planting, even super early planting. Corn, on the other hand, doesn't like super early planting because we want very uniform emergence. We want very uniform emergence and rapid emergence from corn. Soybean has more plasticity. It adapts more to the environment. So even though we might not end up with a final stand of 100, 120, we can still get a lot of good yield with early planted soybean. That was the case for us this year. We planted soybean March 13, we got frost, cold. We only ended up with about 60% of the final stand. We were planting 120, maybe we ended up with 80 to 90,000 plants per acre, and we still got 100 bushels. That's because soybean compensated, it has plasticity. If you plant 38,000 plants per acre of corn and you end up with only 28, it's not going to compensate. You're going to lose a lot of yield. So that's why we see this difference with super early planting. Corn needs rapid and uniform emergence. Soybean tolerates more stress and has more plasticity. However, the later you plant, both crops will lose yield potential similarly. And a lot of the yield gain with planting soybean early has to do with those node numbers and better vegetative growth, more biomass. It's just utilizing better the resources. Another way we can improve node number with better singulation. So I got this image here showing a trial that Brandt did comparing a control to a well-singulated soybean. So soybeans compensate. They compensate based on how much space they have to grow. However, if I plant 2 soybeans right next to each other, where are they going to compensate? To one side only. If I have uniform singulation, they will branch more evenly. With more branching, I get more nodes. With more nodes, I get more seeds, and with that, higher yields. And we actually did a trial comparing fast planting to slow planting. And the idea of doing this trial at the research farm came from talking to Alex Harrell. He says, I asked him, hey, what's the number one thing that you changed from 22 to 23? That was the first year he got the record. What was the number one thing he changed? I was hoping he was going to say, oh, I started utilizing Brandt products. Which he did. He did start using it that year. But he said the number one thing he changed was attention to singulation on soybeans. And how does he get good singulation on soybeans? He plants lower plant population and he plants slower. There's no other way of getting those soybeans well singulated. Those are round seeds. If you're going fast and that trench is open, it can roll on the trench, and singulation is just not going to be good. So after talking to him about that, I decided to do a trial looking at a fast planting speed compared to slow. And this was actually one of the biggest responses we got the farm this year. Is screaming difference just because we got a better distribution of those seeds. So very important for soybean. We want to maximize branching. We want to maximize the number of nodes. We gotta singulate them better. Not only important because of the branching aspect, but also think below ground. we always like to think above ground. We don't think about the roots. If I have two soybeans right next to each other, the moment they germinate, what are they gonna sense? Competition. The only goal for the soybean plant is to produce viable seeds for the next generation. We like the protein, we like the oil, we like the soybean for other reasons, but for the plant doesn't think about that. The only goal the plant has is to produce viable seeds for the next generation. If that seed starts to germinate and it senses, oh, I'm in a highly competitive space with low availability of resources, because those plants can communicate, they know they're in high competition. What are they gonna do? They're gonna lower their seed producing potential, lower the number of pods, because they think, or they know because of high competition, I cannot sustain a high number of seed. And that's true for corn as well. That's why it's so hard when you increase plant population. Why a lot of times you don't see increase in yield on corn, for example? Because we know one of the yield components on corn is number of plants per acre. Why increasing the number of plants per acre doesn't increase yield? Because more plants mean we're competing more for resources. If I don't provide those resources close to the root for it to sense I have enough substrate, I have enough resources for a high kernel number, it's going to lower the kernel number. The same is true for soybeans. So if we don't have good singulation, it thinks it's in a high competitive environment and it lowers seed number production. So there's some above ground but also below ground reasons why singulation is so important. So singulation is one way we can get better branching, but other ways, and I kind of made a list here, of thinking about soybean branching, and that's what we want. We want a good root system. Population, why are we seeing tendencies to decrease planting population? One of the reasons is because it compensates pretty well, it's going to utilize the resources better, and we can singulate better low population. So if you talk to Alex Harrow, his first year he got the record, 78,000 plants per acre. The second year I think was closer to 100, so it's a lower population. People were planting 160, 70, 80 back in the day, so lowering plant populations can help with singulation. Carbon availability and early growth. So the plant is going to decide, should I emit a new branch? Should I start growing more vegetatively based on how much carbon and how much nutrition I have available early on? So starter fertilizer, planter box product, anything that can promote that plant should be more vigorous, and more energetically sound early on can really help with branching. Interaction with genetics, we know that different genetics tend to branch differently, and also hormone balance. So what impedes those lateral buds to start growing? Do you guys know? It's usually a hormone that is controlling that. So the name of the hormone is auxin. So auxin causes in the plant a phenomenon we call apical dominance. So there's a high concentration of auxin in the meristem, and because of that, it blocks those lateral buds from germinating and start growing different branches. Who fights auxin inside the plant is cytokinin. Cytokinin is super important to release those lateral buds and improve branching. And where is cytokinin being produced inside the plant? Most of it on the tip of the roots. So that's why it's so important to have vigorous root growth early on so that we produce more cytokinin. With more cytokinin, we block auxin and we start branching more. And that's also a technique that Alex Harrow uses. He utilizes products in furrow and 2x2 for what? Promote root growth. With more root, we have more cytokinin. With more cytokinin, we have greater chances of branching, and with that, we end up with bigger node number. And here I want to bring a product that is very interesting when you think about promoting root growth. This is a new product we've just released. It's similar to other products in the market. It brings 3 different phytohormones: cytokinin, auxin in 2 different forms, IBA and IAA, and GIBS. Why is this product so good in promoting root growth? So these different phytohormones are controlling root growth. We know that at the meristem here, at the apical meristem of the root, we have a high concentration of auxins. This auxin is telling the root, hey, keep dividing the cells. Divide cells because we want to grow fast. So by applying auxins to the root, we'll promote longer roots because we'll be dividing more of those cells. The cytokinins will be telling those cells, hey, stop dividing, let's start thinking about elongation. So that's what cytokinin is doing. And then GIB is the one who's actually working on elongating and making those cells longer. And then after a while, after that root starts growing, we get into the differentiation zone where the lateral roots grow, and there, again, auxin will be the main hormone driving that lateral root growth. So applying this product in furrow is a very good way to improve root growth. With more roots, we get more cytokinin, we can get more branching on soybeans. I know this is not a soybean seed, but the same applies to a soybean seed. What else can this product do when applied in furrow, octorize? We know that we need gibberellins, GIB, to activate this aileron layer. This aileron layer has the amylase that will break down the starch into glucose and feed the embryo. So if you look at phytohormone concentrations inside the plant, we have a huge spike in GIBs early on so that we start feeding the embryo and we improve germination. So that's another effect of applying this product in furrow. It can really help with seed germination because of the GIBs in it. So that's one product that I would recommend trying out to see if you can improve root growth early on. A second product that is very interesting, and actually maybe that's the reason I'm here, because Blake really liked what he saw when we were showing some plots that we tested this new product. So this is Seaweed Max. It's an Ascophyllum nodosum extract. Ascophyllum nodosum is a brown seaweed. It has a very high concentration of actives, including several phytohormones, including auxins. What are those phytohormones doing? Promoting root growth. So for in-furrow application, we're aiming root growth, and it can also be applied foliar to manage plant stress and soybean architecture. And I'll talk later about managing soybean stress with seaweed, but just to touch on the soybean architecture portion, research shows that foliarly applied Seaweed Max, or not Seaweed Max, Ascophyllum nodosum, blocks the degradation of cytokinin inside the plant. So foliar application of Seaweed Max can also improve branching, root growth for in-furrow, and also branching and stress mitigation for foliar applications. And the reason this product is so interesting because we've been working with seaweed extracts for quite some time, and we identified that there's one component in seaweed that makes it pretty messy to work with, which is called alginates. It's a protein that likes to bind with nutrients and makes it hard to mix with other products. So what we did is we took out those alginates to improve mixability, and we identified the components that were most active, and we have a higher concentration because we increased the concentration of these components. So it's a very powerful, and we're actually one of the only brands that sell seaweed extract and tells how much of these different actives we'll have in the product. So it's a super powerful product, and this is what Blake actually saw at the research farm. We had this product in all of our planting dates. We had 5 planting dates, and we had with or without in-furrow. And the in-furrow was Seaweed Max, a pint of it plus a pint of Enzop zinc. Enzop zinc is a combination of enzymes plus zinc, super important as well. But we were seeing screaming differences, and later on it was even bigger. The difference on camera is kind of hard to see, but Blake was there, and it's...
I've never seen anything like it, and so then I came back and I made Matt sell some, and it was very spectacular foliarly, and then at the end of the day, tracking the yield wise, I mean, it was as consistent as anything I've ever seen. But a little bit of that goes a long ways. It's pretty powerful stuff. But like you said, I don't think there is a product on the market that is that concentrated.
No, definitely
It's
not.
25% brown algae, correct?
That's a good question.
I mean, it's pretty,
it is very concentrated.
I mean,
yeah.
it's like 100 times more concentrate than anything else you can access.
Yeah.
So it does its job. Let's just say that.
It's a good product. I really like this product on soybeans. Corn is good as well. I think in-furrow and corn is better. Soybeans, both in-furrow and foliar. And I'll show some foliar applications of this, some pictures that Blake actually took. It's screaming. Like a week after, you were seeing like crazy differences.
It's crazy.
And this was very visual. We were seeing bigger plants, bigger roots. We were pretty cloudy early on during the spring last year in Springfield. So we were kind of struggling getting nodules and getting that nitrogen fixation going. With bigger roots, we were seeing that we were having more nodules as well because we are growing more roots, we're finding more of those braided rhizobium and having a higher nodule count. So that really helps as well. Yield differences, well, we got some good yield differences depending on the planting date. The first planting date, we actually did not see yield differences. Sometimes you do one management practice and there's so much going on still that doesn't really affect yield at the end. But for all different other planting dates, the yield differences were there. Almost 2 bushels there. Here we got almost 4 bushels on the later planting. So it was still responsive and the yield level was pretty good already. So here's another product. I brought these pictures actually from Brazil. It's Plant Start. It's a product that we have, which is that Seaweed Max plus some phosphorus. So potassium polyphosphate, so it has potassium, so it has some phosphorus, and this is the effect on the root growth. It's very visual, and Seaweed Max does the same thing. So we're improving root growth, improving above bound biomass, and here are some numbers. Look at root mass. It's 2.5 compared to 4.2, and this is replicated data looking at that combination of P plus Seaweed Max in furrow. Another reason or another management practice that we like to do, and this will be impacting a lot on, remember I said carbon availability and overall plant health has an influence on how much the plant wants to branch or not. And managing stress of soybeans after herbicide applications can really improve overall plant vigor and plant growth. So soybeans are tolerant to glyphosate if you get the genetic pack for that. But it doesn't mean that it likes glyphosate. It still has to metabolize glyphosate internally, kind of gets stunted. It stresses the plant. And you kind of see the plant hold on and growing for a little bit. Well, we can manage for that. And that's one of the things, one of the tools that these high yield growers are utilizing a lot. They manage this herbicide application very well. They know until what time of the year they can be applying, until how many GDUs I can be applying herbicides, because if I apply too late, I'm damaging that yield potential too much. And they always like to pair herbicide with nutritionals to help metabolize. And that's what I want to explain here. So when any stress or a herbicide application occurs, the way the plant will signal that stress is increasing ROS production. What is ROS? ROS is reactive oxygen species. This is natural. The plant has a way to cope with those reactive oxygen species. But a herbicide application is a very strong event for that plant. So we suddenly have a surge of reactive oxygen species. This can cause damage to DNA, lipids, proteins. It's not good. It bursts the cells. This is how contact herbicide kills weeds. It makes that weed produce a lot of reactive oxygen species. It kills the plant. Every plant produces ROS, but we have a way to deal with it. But when we apply a herbicide, it increases so much that it stresses that plant, and then you start seeing some symptoms of that stress. Well, how are these plants managing these ROS? The main enzyme they're utilizing is called superoxide dismutase, SOD. That's the main way the plant cleans up ROS. It converts reactive oxygen species into hydrogen peroxide, then we have catalase, peroxidase, a lot of stuff that will continue to break it down into CO2 and water. The key here is that there's 3 different types of SODs inside the plant. One of them is activated by copper and zinc, a second one is activated by manganese, and the third one is activated by iron. We have a product that has zinc and manganese, 2 of the most commonly deficient micronutrients. These applications will activate the superoxide dismutase and reduce stress caused by this herbicide application. So these high yield growers like this product so much that what do? They apply what they call a primer pass. They come out like 2 or 3 days before they apply a herbicide. They load that plant with Smart Quattro Plus or Smart Trio that has zinc and manganese. Prepare that plant to metabolize that herbicide the fastest it can. Because the fastest that plant metabolizes that herbicide, the faster they start growing, the more energy it has to branch and produce more nodes and seeds per acre. So that's why we always like to pair herbicides, and that's pretty common in central Illinois, in the area we're at. Smart Quattro Plus or Trio with your herbicide application. What's the name of the product you had?
It's complete. So we would have Smart Quattro as our base. We can also access Smart Trio, but then we also, in that Smart Quattro, we have L-proline and phenolic acids in there.
Yeah.
So just a little bit more, he's gonna talk a little bit more about stress mitigation.
Yeah, yeah.
Very, very, very similar to what they're trying to accomplish here, and like I said, we can access Smart Trio, both excellent products. On your data, have you seen anything different? Now, I know a lot of guys aren't gonna go do this, but if you preload that plant before, like 3 or 4 days before you spray that herbicide, are you seeing big yield differences?
Yeah, that's one trial we want to have for next year. We actually already have an area on our field that will test that out.
Okay.
Because we've been talking to these high yield growers, we never tested it to see if it actually makes a difference with this primer pest or not, but they swear that it makes a difference. So
Okay.
they put it as a primer
Yep.
and with the herbicide. So
Yep.
both times they're coming in with the nutritional.
Yep.
And a lot has to do with these enzymes to deal with stress. And I'll talk a lot about stress, and the key thing is these reactive oxygen species. We gotta manage it. It's the same thing on your body. Everyone here has reactive oxygen species on our body. When you're feeling a little bit down, sick, don't you take like vitamin C?
Mm-hmm.
What is vitamin C? An antioxidant is reacting with reactive oxygen species in your body as well. Same thing with plants. We in our blood have superoxide dismutase as well. So it's very similar, plants and humans. And we are learning how to manage for these reactive oxygen species because they're one of the main causes of yield loss. And I'll explain why we're losing so much yield from stress. Another reason why we like to apply this nutritional with our herbicide is because we're usually applying this herbicide around these V stages. It depends when the plants start flowering or not. Right before we get in peak nutritional demand. So this is a graph that I did during my PhD at the University of Illinois. Dr. Penny showed you guys that accumulation graph, where it's going, grain, stalk, leaf. So I had access to the original data set because those graphs were done at the lab that I worked at, and I converted from cumulative throughout the growing season into rate of uptake. What is the difference? Here I'm looking at pounds of nutrients per acre per day. How much nutrition is that plant requiring on a daily basis? And this is where it aligns very well with what Dr. Penny was saying. Wasn't he talking so much about like concentration of those nutrients and why concentration is so important? Well, there is one correlation between concentration and rate of uptake. The higher your concentration, the higher the rate of uptake of these nutrients. The higher your yield, the higher the rate of uptake will have to be. So that's why concentration is so important. But managing concentration of micronutrients in the soil, it's a lot of time hard because micronutrients are super reactive. They want to precipitate, think with me, iron, copper, manganese, zinc. All of these nutrients are what we call transition metallic nutrients in the periodic table. What are transition nutrients? They either want to donate or receive electrons. They're super reactive. So if you leave like a cast iron pan out on the weather, won't it get rusty? It's because it's reacting with water, oxygen, it's precipitating in the form of iron hydroxides. That's the same is true in the soil. If you apply iron to the soil, it's going to precipitate. If you apply zinc to the soil, it's going to precipitate. If you apply manganese to the soil, it's going to precipitate. That's why you apply zinc EDTA. That's why I apply zinc DTPA to protect it. So managing micronutrients through the soil is not that easy because you got to have it chelated. Those chelates don't last that long, so we don't have season-long availability. That's why applying these nutritionals through the leaf is so good, especially for micronutrients. That's why we're managing so much of our micronutrients through the leaf, because managing only through the soil is not that easy. Does it work? It does. We love putting some zinc EDTA in fertile. We love putting some manganese EDTA. It works, but it's not season long. And then you complement through the leaf at certain specific stages where requirement gets so high that the soil might not be able to keep up with plant demand.
Are you going to talk about smart technology?
Yes.
That's what I heard just now, too.
Yes, I will.
Yep.
I will right after this, actually.
Yep.
Here I'm going to show a graph. We have 5 years of data here from 2019, 2024, 33 locations, different products in our smart system technology. Most of them are Smart Quattro Plus, but some Smart Sulfur Plus as well, Smart Manganese Plus, very similar products that Blake has. And the responses are just great. It's very rare for us to see negative results from this. So this is a pretty standard for us. adding a nutritional to your herbicide pass. On average, we are seeing 2.5 bushels to the acre, and this is all third-party replicated data, universities, CROs, all of that. And here's where I want to talk about smart system, because it's not as easy as just go out there and apply any form of nutrient. I just talked about how reactive these micronutrients are. If they're reactive in the soil, they're reactive in a tank mix, they're reactive when they get to the leaf, they don't move through the leaf. So we gotta add technology to what? Kind of like give it a hug. You give it a hug, stop making it react with anything else, improve uptake, mobility, and utilization by the plant while minimizing reactivity with other components in your tank mix. So smart system it's not necessarily a product or not necessarily a carrier system. Smart system are some standards that we gotta meet to make this product in our smart system line. And the standards are very good tank mix compatibility. It pretty much mixes with everything. No herbicide antagonism, that's one of the things we were seeing a lot. You mix your manganese product with your glyphosate and then the glyphosate doesn't work that well anymore. So we want to make sure that our products are not causing any antagonism and very good efficacy of uptake, movement through the plant. Also, wind tunnel testing, volatility testing, we do all of that. And we do all of these tests prior to launching a product because we want to make sure that the grower doesn't have any struggles when he has the products on his hands. One important thing that you guys gotta remember is that leaves are not made to absorb nutrients. There's no zinc in the air, there's no manganese, there's no copper, there is no nutrients on the air. So the leaves were not made for that. Leaves were made to protect that plant from dehydration. For that, it created a very thick, waxy cuticle that impedes dehydration. It created some stomata that opens and closes and control movement of water and CO2 and oxygen. And it's made as a solar panel. I'm here to absorb sunlight, not nutrients. But with the right formulation, you can make it possible. And that's why this formulation is so important. We need a formulation, as I said, we have complexing agents. Not EDTA, because EDTA is a very large molecule. It does provide very good tank mix compatibility, but it's such a big molecule that it doesn't pass through the leaf, the epipores. Our complexing agents saturate those nutrients, improve absorption through the leaf, improve mobility inside the plant to the new growth, and also we have some humectants, some sugar alcohols that keep that nutrient in solution for longer. Because zinc doesn't have legs either, so if that solution dries, it's not going to get into the leaf. It needs to be in solution. So if you don't add technology, if you don't add humectants, if you don't add complexing agents, absorption and utilization by the plant is very low. And we've been doing this for over 50 years. So we started our micronutrient business back in the early 70s. So we understand a lot about formulating with micronutrients. And this is one of the coolest technologies that we now own. We're actually one of the only companies in the world that, we are the only company in the world that has an equipment like this. So this is what we call micro XRF. It's pretty much an X-ray of the leaf, and we can see how these nutrients are being absorbed and moved throughout the leaf. So here is where we applied zinc, smart zinc, and here's the area of the leaf that we're scanning utilizing this micro XRF. So everything in red that you see is zinc moving to the phloem and moving inside the plant and being absorbed. So we own one of these equipment, and with that we can look at how to improve our own formulation, how our formulation is working compared to the competition. So it's a pretty neat equipment. It's pretty expensive and it's kind of hard to work with it. We actually just bought a newer version that is much bigger. So here we're only able to scan a small portion of the leaf. Now we're able to scan the entire leaf. It takes almost a day to generate one of these images, but it's pretty neat. And here you're looking at Maniplex, a different formulation we have, comparing it with different additives. So this is how much we're investing to actually learn and see how our products are performing. Now we can convert from this into numbers, and this is research done by Patrick Brown. He's one of the world leading experts in plant nutrition out of University of California. And he was comparing our formulation, Smart Zinc, to smart or zinc sulfate, the salt form of it. What is he looking at? Permeation rate. This is the most limiting step for nutrient absorption, passing through that waxy cuticle. Wax doesn't like water. How are those nutrients passing through it? It's very complicated. So here we're measuring how much of that nutrient is passing through the cuticle over time. You can see that we have faster absorption with Smart System, and we have much longer absorption. And this has all to do with the humectant properties. We keep that nutrient in solution for longer, keeping that plant absorbing it. Here's a different way of looking at it, and this is very important as well. So when you guys go out there and spray herbicide or fungicide, what GPA usually you're applying? 15, 20? Do you get that entire plant covered with solution, with the spray solution? We would love to, right? But usually it doesn't happen. So if I have a leaf and only certain droplets fall on that leaf, if that nutrient doesn't move laterally, if it doesn't diffuse laterally, only those cells right beneath that droplet are going to get the nutrients. So we're going to have low utilization of those nutrients. What are we looking here? We're looking at the lateral movement of those nutrients. So the methodology also done at the University of California, we applied the nutrient right at the center here, and we collected discs every 2 millimeters, and we calculated how much of the applied zinc moved laterally to those discs. So with zinc sulfate, 80% stayed at the application point. 20% moved laterally, but only 6 millimeters. So it did move a little bit, but it didn't move very far. With Smart System technology, almost double the amount moved laterally, so almost 40% moved laterally, and it moved much farther away. So we have much better utilization by that plant of those applied nutrients. So not only getting through that waxy cuticle, but also moving laterally, super important in this case. So here, those were the points I wanted to talk about on node number. Now we're back to the yield components, and now we're going to talk about pods per node. Any questions on node number or the different practices that we talked about? Does it make sense or not really?
See, me and Matt always talked about stress mitigation. Now we heard it from the PhD himself. So,
Yeah.
so we were right.
Yeah. And
Well, it's PhD.
And we'll talk even more about stress mitigation now, because soybeans will abort from 50 to 75% of all flowers in small pods. And the main reason they're aborting those flowers in small pods is because of stress. 2 types of stress: abiotic stress or stress caused by nutritional deficiencies. And those are the 2 things that we want to address here when we talk about pods per node. How to improve pods per node. One of the key nutrients that we are seeing that really helps improving pods per node is boron. Boron is another micronutrient that is very hard to manage through the soil. Soil, guys, is always your main source of nutrition. So I'm not saying boron in the soil is not important, but it's hard to manage it season long because of how mobile it is. So boron in the soil is found in the form of boric acid. It doesn't have any charges. So if you think nitrate and sulfate leaches a lot, boron leaches even more, because it doesn't have any charges to be attached to that soil. So if you apply boron early on, it's very likely that you won't have enough during some key stages for the plant. And for soybean and corn, right prior to tassel or right prior to R2, full flowering is when we need most of our boron, because boron is so important for pollination success, pollinic tube growth. There's a lot of boron that goes into those flowers. And if you think about all the different micronutrients on a molar basis, not ounces, not grams, molar basis, what does that mean? The amount of molecules of those micronutrients. Boron is the one required at the most. But because boron is so light, the amount of ounces you need is not a whole lot, but on a molar basis is required the most for soybeans, because dicots need a lot of boron because of how many flowers they produce. They produce a lot of flowers need a lot of boron, so we have a huge requirement for boron. And boron is likely to be deficient. So foliar applications at full flowering or at R3 really help with increasing flower and pod retention. With that, we get greater seed numbers. So this is a micronutrient that is key for us in our management for high yield. And boron is not easy to be applied through the leaf either. The reason is over 90% of all boron in the plant is found on the cell wall. It combines with the pectins and it kind of strengthens the cell wall. So you go out and you apply 10% boron or boric acid. The moment it touches the leaf, it gets stuck to that cell wall. Why? Because it didn't have that hug that I told you guys, that complexation. So we have a way of complexing our boron that mimics the cell wall. So it passes through the cell wall and has better lateral movement and diffusion. So we have a specific formulation for boron that is in our boron technology that is super, super efficient for foliar application. And the product that we usually pair with our fungicide at these stages during flowering is Smart BMO. What's the name of the product you have, Blake?
We call it
BMO, yeah, it's a great product. So Quattro Plus and Trio with your herbicide and fungicide with your BMO Plus Trio doesn't hurt, but it's a great product. It really, really helps. And not only it has boron, but it also has molybdenum. And molybdenum on soybeans is key.
especially here, because we hold a ton of nitrate in our soils. And we typically plug up that plant with nitrate. You can look at all of Beck's PFR data in our regions. Smart BMO was the highest ROI in these areas.
Wow. Yeah. So moly, super important for nitrogen utilization in the plant. For corn is important because of nitrate reductase, but on soybean is even more important because it activates nitrate reductase, but also nitrogenase. So where does soybean gets its nitrogen from? Soil and the air, usually half-half. Here, maybe even more from the soil than the air, but usually half-half. To fix the nitrogen from the air, we need an enzyme called nitrogenase. Moly activates this nitrogenase, is a cofactor for nitrogenase. So super important to utilize nitrogen from the air. And moly also activates nitrate reductase that convert nitrate that your plant absorbs from the soil into nitrite, and then ammonia, and then amino acid. So the plant cannot do anything with nitrate. It doesn't do anything. It has to assimilate it. And to assimilate it, we need moly. So here we're combining boron for pollination and reproductive success, plus moly that improves nitrogen utilization. And moly is so important at these stages because when we're applying here our fungicide in our 1 to our 3, this is our nitrogen requirement for soybean. From planting all the way to our 8 pounds of nitrogen per acre a day, we are at peak nitrogen demand. What happens if the plant doesn't get enough nitrogen that it needs to sustain all those pods and flowers? It's gonna abort. So 2 things here. We're providing boron plus moly that improves nitrogen utilization and decreases abortion because we have better nitrogen status in the plant. A lot of people don't talk about nitrogen on soybeans, but nitrogen on soybeans is even more important than on corn. For every bushel of corn you produce, you need about a pound of nitrogen. For soybeans, it's about 4 pounds of nitrogen per bushel produced, so 4 times more. But we get half of it from the air, half from the soil. We don't need to be fertilizing as much, so we don't think about it. But still, it's super important. And it's likely one of the most limiting nutrients for high yielding soybeans. And I'll talk about it later on, especially during grain field. But at this stage is also key because of the high requirement. And one graph that I forgot to add to this presentation, but really tells a lot, is this is for 60 bushel soybeans. So this was done at the University of Illinois in 2013, almost 10 years ago. But if you increase yield to 80 bushels, what changes? It's mostly during this peak, so we have a greater requirement. So the higher your yield potential, the more nitrogen you require on a daily basis, the more important that moly is going to be. That's why I always say, you're more likely to see a response to these products and this foliar application biostimulants if you have your system all working pretty well, because then your yield level, your yield potential is so high that you need those supplementation, you need that help applied through the leaf. If your yield level is low, let your soil do all the job. You don't need to be applying Smart Trio, you don't need applying Smart Quattro Plus, but the higher your yield potential, the more need there is for supplemental fertility and the more important foliar applications are going to be. We've done a ton of testing. This is one trial, a very interesting trial, actually several trials, 54 total evaluations. So we did this with Dr. Fred Billow at the University of Illinois, 3 different regions in Illinois. So central to northern Illinois would be similar to here, moldy soils, high fertility. Southern Illinois is alphy soils, lower organic matter, so different type of soil. So we had 3 locations, 3 different years and 6 replications. We looked at Smart Trio plus Smart BMO FR3. And this is the average yield increase on top of what the fungicide was already giving us. So this was applied with a fungicide. So adding this nutritional to your fungicide can really, really help improve your yields. And we've done testing elsewhere as well. This is 38 locations over 5 years. Average yield response of 2.9 bushels. So here we're looking at different products in our smart system technology, but most of them are Smart BMO plus Trio, the red bars. We also have Smart KB, which is Smart BMO plus a pint of potash. So if you're lower in potassium, if you need to improve your potassium utilization, Smart KB is a great product as well. So a lot of testing and a lot of positive results with these products. Moving on, we talked a little bit about nutrition, how to retain more pods per node. Now I want to talk about how to manage stress directly, utilizing some biostimulants to reduce pod abortion, to reduce flower abortion. And I came across this paper. This is a paper that was actually published the end of last year, in 2025. And I came across it. It's a paper talking about plant developmental decision to either abort a flower or set seed. So what is happening inside the plant for the plant to decide, hey, I'm going to keep this pod or I'm going to abort this pod? And some key things that they brought in this review are ROS accumulation and cytokinin levels. When we have ROS accumulation caused by stress, that's a signal for the plant to abort flowers, abort small pods. And also cytokinin balance. When cytokinin levels go low, we start to abort flowers and pods. So a lot of what I'll talk about here is either managing those ROS or managing that cytokinin. And the first product that is very interesting is also a new product, and it's a product that Alex Harrow used pretty much every week on his high-yielding beans, is OptaThrive. And OptaThrive, it's a UPA-registered PGR. It has cytokinin, it has GABA and choline chloride. GABA and choline chloride are acting directly on mitigating plant stress. Cytokinin helps with plant stress, but it helps also retain flowers and stay green and fat. And I made this pretty nice diagram trying to explain, summarize what this product is doing. So greater stress resilience, that's coming from GABA and choline chloride. So choline chloride is a precursor for a phospholipid. You guys remember from biology classes, every cell has a biphospholipid membrane. This biphospholipid is produced by a precursor from choline chloride. So those reactive oxygen species can break these membranes. We need choline chloride to repair it and make that cell function properly. So that's what choline chloride is doing. It's also a precursor for beta-glycine, something like that, which provides osmolar protection against dehydration, too much heat. Pretty common to be above 86 degrees here during the summer. In Illinois it is. Pretty much every day during the summer, 95, 98, almost 100 degrees. Soybeans are C3 plants. If it's above 86, it's no bueno for these plants. They don't really like it. So a lot of the environments we're growing these beans, it's pretty much a stressful environment. So we need to be protecting against stress. They're going to happen regardless of the season you grow them in. GABA, super important for antioxidant defense. Remember the SOD, remember that peroxidase that controls the ROS. So yeah, GABA activates it. It's also important for pollen guidance. So the pollinic tube has to get from the pollen to the ovule. How does it guide itself? By difference in concentration in GABA. GABA is doing it. So GABA is super important for pollination success as well, and it accelerates recovery after stress. And this is the nerdy slide that I want to show you guys. A lot of the slides are nerdy, but either way, this really tells the story of what GABA is doing.
I was just
inside
gonna
the
say,
plant.
I want everyone when they're spraying to think of this. These slides.
But it helps you understand why you would use the product.
Good.
The yield results are there. I could come here and just tell you yield results, but if you understand how it's happening, it's easier. So the plant absorbs nitrate and ammonia. As I said, the plant cannot do anything with nitrate, it cannot do anything with ammonia. It has to assimilate these nutrients into amino acids. In good growing conditions, it becomes glutamate, and then it becomes amino acids. In stressful conditions, what happens? The plant naturally converts glutamate into GABA, and GABA does all of that we talked about to improve resilience against stress. The key thing that I want to bring to you guys here is you go out and you apply Optatribe. Stress doesn't happen. Well, the key thing here is that GABA is not a wasted product. The plant naturally recycles GABA into carbon and nitrogen. So we're not wasting that product. It's going to be utilized for biomass and nitrogen utilization inside the plant. So very interesting product. It's a new product. We don't have a ton of data with it yet. We're going to start collecting more data. But this is a product that we would recommend applying from R1 on. During reproductive, all the way to the end of the season, you can be applying it. It's a super, super interesting product. The second product that I want to talk about, and I know that Blake likes this product, is BioFlex. So BioFlex is a 3-in-1 product, also focusing on stress mitigation inside the plant. And this is a 3-in-1 that contains an adjuvant. So we usually apply BioFlex with post-herbicide. So if you're gonna add an NIS to that post-herbicide, BioFlex already has it. And it's at a very competitive price point. So it has a good adjuvant on NIS, AP and PE free. It has phytosterols and fulvic acid, and I want to explain how these actives are working inside the plant. So we have a fulvic acid, which you guys talked about with Penny. We like fulvic acids. We like fulvic acid in furrow. We like it on the soil, but we also like fulvic acid applied to the leaves. Why? The fulvic acid we work with has a very high concentration of phenolic acids. And we identified these phenolic acids after working for several years with these concentrated actives here. And we found out that they're very good in mitigating stress. What are these products doing? They're upregulating the immune response inside the plant. So they upregulate the genes that produces those enzymes that detoxify the plant inside it. So why is upregulation? Real quick, just so you guys understand what I'm talking about. So this is how the plant produces enzymes and proteins. It reads the DNA, converts an mRNA, and then it produces enzymes. Upregulation means we'll have more transcription, we have more RNA, and we have more proteins, we have more antioxidant enzymes. So that's what the phenolic acids are doing. We also have encapsulated phytosterols. Encapsulated phytosterols activate this reactive, or this SOD, these enzymes responsible for neutralizing this stress. So the two components here are working against plant stress in a very potent way. And why don't you see more phytosterols out in the market? Well, these actives were discovered back in the 70s, and it's super hard to formulate with them. Very hard. They don't like to be in solution. Brian Heschmeyer and Lucas Lovelace, both chemists from Brandt, they found out that you could encapsulate it. It's a nano encapsulation where you put the phytosterol, this is the phytosterol molecule, inside a beta-cyclodextrin grain, which is sugar grain, nanomolecule, and then it becomes soluble. So that's how we made this product to be soluble and active. And it's a super interesting product. Here's some research results. Back in 24, at the research farm, controlled 108 bushels to the acre. So you're like, yeah, that's probably not a very stressful condition, right? Already yielding 108. Well, you cannot forget that high-yielding crops are always under stress. It's just like a car going 150 miles an hour. Everything is under stress. Just like a high-performing athlete, his body is under stress. A high-yielding soybean is under stress. So that's why we saw the type of results we did. We saw almost 6 bushels when we applied BioFlex at 1 pint per 100 gallons. So the rate is per 100 gallon because it's registered as a surfactant. Rates go from 1 to 2 pints per 100 gallon On soybeans, what we've seen over the last 2 years and all the research we've done, 1 pint is better than 2 pints. On corn, 2 pint is better than 1 pint. We actually just got back our research book from this year, or we just got done with our research book this year. The average response on corn was almost 9 bushels for the 2 pint per 100 gallon over all the different universities and trial cooperators we work with. But either way, very good response to Bioflex. Here we also have Seaweed Max applied foliarly, very interesting results. And here we have a combination of folic acid, seaweed extract, plus nutritional as Biolex. There are also good responses on those products. This is a multi-state trial summary. We did several different states in 2025 with CROs and different universities looking at Bioflex. Unfortunately, we had these trials done at 2 pints per 100 gallon. I wish we had this done at 1 pint per 100 gallon because on soybeans I know it's a better rate, but I don't know why the protocols came out and they were done at 2 pints per 100 gallon. But still, we saw 1.3 bushels yield advantage. Smart Quattro Plus on average 1.2 bushel. Usually we would see a better response than that, but this is data, I can't argue with data. And the combination of Bioflex plus Smart Quattro Plus 1.6 bushels to the acre. This is a trial we did at the research farm. 3 replications looking at Bioflex at 2 different rates. 1 pint per 100 gallon will be about 3.2 fluid ounces to the acre, depending on the GPA you're running, but we did it at 3.2 fluid ounces to the acre, with or without Trio. 2 pint per 100 gallons, 6.4 fluid ounces, with or without Trio. And also a combination of this was applied all at V4, a combination of V4 plus R3, getting in with just Bioflex or Bioflex plus nutritionals. So what did we see? Remember I said, I think the lower rate is better, and this is what the trial showed us. The 1 pint per 100 gallon gave us a bigger yield response, almost 2.2 bushels to the acre. When we added Trio, it actually brought it back a little bit. And now we have a new theory. We saw this across different trials that we did. Trio is a very acidic product, different than Quattro Plus. Quattro Plus, it's a more alkaline, not alkaline, it's buffered around 6, 6.5. Trio has a pH much lower, around 3. We think that Trio is too acidic and was making Bioflex a little bit too hot, if that's a fact. We're not sure. We did not see a problem with Quattro Plus, but we did see a problem with Trio. The higher rate of Bioflex did not give us the yield result we wanted. With Trio, kind of mixed there. And here, only the combination of nutritional was very responsive, but only Bioflex applied twice was not very good. And again, here we'll have a total of 6.4 fluid ounces. And that's when I bring this graph. I got it from Dr. Sybil at the University of Illinois, where he really likes to use this graph to talk about most biostimulants. Most plants are pretty sensitive to these molecules, especially phytosterols, which are plant hormones. You've seen how sensitive soybeans are to dicamba, for example. That's an auxin. Phytosterols are also hormones. Our plants are very sensitive. So applying too much doesn't mean it's going to be better. So keep it to the label rate. So soybean probably better. The lower rate because the plants are very sensitive to these actives. Here, trials done in 2024. I have marking here Seaweed Max, but also Bioflex. Bioflex in 2024 in multi-state, we saw 2.4 bushels to the acre. I think the reason we ran with the higher rate in 2025 is because 2024 they did look very similar. They were on average the same response. 3.4 and 3.2 and 6.4, but in 25 we've learned that probably the higher rate is not the best. But I wanted to point out Seaweed Max, because Seaweed Max applied foliarly gave us excellent results in 24, gave us excellent results in 25, and it's a product that can be very much utilized for this foliar application. And these are some pictures that I got from Blake. He was looking at herbicide only. Here he has herbicide plus Seaweed Max. You're seeing much less phytotoxicity. It really increases the plant resilience to stress. It can work with your herbicide application. Again, this is what it's doing inside the plant, is increasing also production of this superoxide desmutase. That's what Seaweed Max is doing, and also these other enzymes that are breaking down. So we know from the literature that seaweed extract is very good in detoxifying the plant. Also, if you're seeing bigger plants and you're seeing it kind of branch a little bit better depending on the soybean. Why? Again, seaweed extracts can block the degradation of cytokinin inside the plant. They can promote plant growth, vigorous plant growth. So very interesting response here to foliar seaweed extract. And here's Seaweed Max plus S17. What is in S17 again, Blake?
So it's just it's 8% nitrogen, 17% sulfur.
Yeah.
So
How long after application was this picture?
what was it, like 14 days?
Something like that.
Yeah.
So one thing I did notice, at least locally around here, when we ran, we pretty much paired Seaweed Max with the foliar nutrition, whether it was that or Smart Kid B or BMO or Quattro Plus. We saw it almost double yield compared to when we were running it by itself.
Wow.
And we're assuming, and you had a theory behind it, Marcos, is that, I mean, we were stimulating so much, we created a lot more opportunity to fill stuff out, so I think we just maintained it more. So we had so many more fruity bodies on that.
Exactly. You're stimulating, you're improving growth. With more growth, you need more nutrition, so you're more likely to see a response from the
What stage was that applied at?
Oh,
This was herbicide.
the one thing I do like about Seaweed Max is it feels like you don't have to be stuck to a certain time frame, whereas like hormones, there's certain early season hormones that you talk about, there's some late season hormones. I know Matt works with another product, Pixie Dust Plus, that's more specific timing. This is a little bit broader, and it's fairly economical, and it kills a lot of things. It checks a lot of boxes: stimulation, mitigation, overall nutrient use efficiency. And so I couldn't even tell you when this is sprayed, and honestly, I didn't follow a specific protocol, but it just seemed like post herbicide spray pass whenever we got there. I mean, it was very consistent with what we were seeing in responses.
Yeah, absolutely. It's a great product, and can run in furrow.
Yeah, you can run it. It's got a broad use.
Yeah, yeah. So yeah, I was pretty impressed. I heard you. Yeah, you told me right after you got those pallets from Seaweed Max, you were seeing all these different crazy responses.
Yeah,
I was pretty excited about it.
We just basically tested it last year. I mean, and this year I think we feel very comfortable. Brandt has 2 years of data behind it. Our product, we call it, it's private label, it's called Sea Surge. It's the same exact product. But,
Cool.
and that BioFlex you talked about, Dr. Penny was talking about Fulvix and Humix. That BioFlex on corn, I mean, that's a pretty heavy dose of Fulvic in there. I ran some trials with that this year at the 6.4 ounce rate with fungicide timing. It's a little bit more expensive, but I mean, we were seeing a little over 10 bushel response. You said your data was around 9?
Yeah, average of 10 locations, around
Yes.
9 bushels.
Yes, so very, very similar to what Marcos was seeing there. So that's good that kind of matched up. Soybeans, I didn't get a run it on. I kind of was hoping I was going to keep Seaweed Max as my soybean product, and I was going to do the BioFlex, or we call it BioThrive, same product, on the corn.
Yeah, that's what I would
and we experimented with different rates, but it does appear the 6.4 ounce rate is better.
Yeah, so yeah, you're doing exactly what I would do. Very, very good product on soybean, this ascofilm noodles here. Alright, I don't have that many slides anymore. I know you guys are probably tired of hearing me talk, but we've covered all the basics here on seeds per acre. Talk about node number, pods per node, how we can improve that. Now I want to talk about weight per seed and how we can manage to improve that. Some limitations, some things we think might be the future, but we might not be there yet. So again, why do we have to manage for weight per seed? Because we have this negative correlation. So think with me, we've done all this nutritional work, we've done this stress mitigation work, we retained a lot of pods, and we made a lot of nodes, and we have a lot of seeds per acre. If we don't manage later on to improve seed weight, we can not get the yield advantage that we wanted. And if you increase your seed weight from the average, remember average seed weights around 150 milligrams. If you increase only 2 milligrams from 150 to 152, you already get 1 bushel to the acre. That's huge. How can we improve from 150 to 152? We need to do photosynthesis. That's the only way I know that the plant produces sugars to improve seed weight. And here's a trial done in 24 in the research farm, just to kind of showcase it. A systems approach. You start early, but you don't end early. You go all the way to the end, and the gains we can get from that. This is fungicide plus Trio plus BMO. No foliar, one application at R1, 2 applications R1 plus R3, 3 applications R1, R3, and R5. A staircase here, adding yield each entrance that you get into the field. And which nutrients do I want to focus the most later on? The nutrients I want to focus the most later on are the nutrients that we require the most inside the grain. Why is that? Because if we require those nutrients in the grain and we don't get it from the soil, the plant is going to get from the leaves. The moment it gets from the leaves, we lose photosynthetic capacity. So what are the nutrients that we need the most inside the grain? So here you have production coefficient of nitrogen, phosphorus, potassium, sulfur, zinc, and boron. What is production coefficient? The amount of pounds of nutrient that I require per bushel produced. Here I have the removal coefficient. What is the removal coefficient? The amount of pounds that I remove from the field with each bushel that I produce. Here you have the harvest index. The harvest index is the relationship between removal coefficient divided by production coefficient. So 3 divided by 4 is going to be 73%. So from all the nitrogen that the plant takes up, 73% of all of it ends up inside the grain. For phosphorus, 81 of all the phosphorus that the plant absorbs from the entire cycle. Potassium, not so high, 45. Sulfur, 58, pretty high. We have sulfur being absorbed all the way to the end of the season. Zinc, 42, boron, 38. So key nutrients that I would pay attention are nitrogen, phosphorus, and sulfur. And how much is accumulated after R4? Remember, we're managing our soybeans until R3. Half of the season pretty much still has to happen. Half of this nutrition still has to be absorbed from R5 on. So if we don't get it from the soil or through the leaves, we are going to remobilize from the leaves and then we're going to lose photosynthetic capacity. So this is a trial we done with that in mind, kind of thinking, how can we manage late season application of some of these key nutrients to improve soybean seed weight. We did it at 3 different locations, and here you have the average result. So the timings, some products were applied only at R3, R3 plus R5, R3 or R5. So what are we looking at? Trio plus BMO, boron, super important for sugar movement, also important for nitrogen utilization. And remember, 73% of the nitrogen ends up in the grain, so we need good nitrogen utilization. We have power phos that has some phosphorus in it. 81% of all phosphorus ends up in the grain. We also have some sulfur in it. We have some sulfur in the other products. So that's what we were looking at. What was the response we saw? So with this, going later, that's where we saw the biggest response. Going in at R3 and R5 with these products, we saw a 5.1 bushel response. Pretty good response. We tried this again, we did see similar response. So we think we're getting somewhere. These nutrients are super important there. Here we have another example looking at Trio plus KB that has the moly plus boron, super important for the sugar movement, plus potassium. We're looking at different plant populations in season long. What is the impact of that season long management? Meaning 1 pass at R1, 2 pass R1 plus R3, 3 pass R1, R3, and R5. For all different cases, other than this population here, we saw an increase when we go season-long management with those nutrients, slowing down remobilization. And here you have the average response from those applications. The key thing that I think is happening here is we're slowing down nitrogen remobilization. So most of these products, pretty much all these applications have moly in it. Moly is super important for nitrogen utilization. And when the plant starts to remobilize nitrogen from the chlorophyll into the grain, we lose our photosynthetic capacity. And here I kind of want to get into this nitrogen dilemma. It's something that I've been thinking a lot about. I don't know if I have the answer yet, but you guys might be able to help me here. So I really think that when we get into grain field, we're short on nitrogen, and that's one of the main reasons why we're not able to increase seed weight. So where are we getting nitrogen from? As I said, half of it is from the soil, half of it is coming from the air. Soybeans need to accumulate 4 pounds of nitrogen per bushel, only which half of it comes from biological fixation, And what happens is, when plant starts filling seed, after R5, the seeds start to become the main sink for photostimulants. The moment we start filling seeds, we stop sending energy down to the nodules and the nodules die. So this is what happens. So nodulation is not a cheap process for the plant. There's actually some research showing some super nodulating varieties of soybean where they have like hundreds of nodules, like 300 nodules. And you would think, man, this plant is gonna do very good. Well, it does not at all, because those nodules require so much energy to stay alive that the plant doesn't grow at all. What happens at R5 when we start filling seed? Our nodulation suddenly drops down. So here you have nitrogen assimilation rate, meaning kilograms or pounds of nitrogen per acre day, from the air and from the soil. So at the beginning we don't have nodules, so we don't have anything from the air. Most of it is coming from the soil. The moment nodulation starts, the air becomes the main source of nitrogen for that plant. However, when we get into R5, that suddenly changes. Why? Again, the seeds start to fill, the seeds become the main sink of photostimulants. We stop sending photostimulants down to the roots, the nodules die. Then we're relying solely on the soil. And a lot of time, it rained a lot in June, July, we lost a lot of our nitrate in the soil, and we don't have a lot of nitrogen to fill that grain. And then what happens? We start remobilizing from the chlorophyll. That is what happens in the fall. Why do the trees lose their leaves? They're storing nitrogen from the chlorophyll into the roots so that they can use that nitrogen for the next season. Here, the plants are taking that nitrogen from the leaves, from the chlorophyll, and putting it inside the grain because they don't have enough in the soil. And that's what happens. This is research done at the University of Kansas that shows the same thing. Number of active nodules throughout the growing season. At R5, suddenly it drops because of that lack of energy going down. And at R5, what do we see? So this is the graph coming from the University of Illinois. Nitrogen uptake. Where is it going? The leaves, stems and petioles, flowers, and grain. We get to R5, when we start forming our grain, what happens to the nitrogen in the leaf? Starts going down. We lose our photosynthetic capacity because of how much we're requiring inside the grain. And this graph really shows what I'm trying to say here. This was done at the University of Wisconsin, looking at 3 yield levels for soybeans: low, medium, and high. This is nitrogen assimilation or nitrogen uptake. Kilograms of nitrogen per acre per day or pounds of nitrogen per acre per day from planting all the way to maturity. Where do you guys see the biggest difference in nitrogen uptake? Early season or late season? Mostly late season. Past what? Past R4, R5. So we see that a high-yielding soybean has higher requirement for nitrogen during grain fill. That's what is explaining the differences in yield. So if we don't have that nitrogen late season, we lose yield potential because we're losing potential to increase seed weight. So how can we fix that? Well, we either going to improve the soil or the air. Let's think about improving the soil nitrogen availability. Is that easy? Can we do that? Well, we can. There's different forms of doing that. I think there's some very good ways of doing that, like managing residue. Corn residue immobilizes a lot of nitrogen. So if you manage that residue so it doesn't immobilize as much nitrogen in the next season, that can be a good option. Soil health, turnover of nutrients, or maybe even apply nitrogen fertilizer. The problem is, the higher the nitrogen fertilizer rate you apply, the lower the amount of nitrogen fixed by the plant. So it's not easy as, oh, go out and apply 150 pounds of nitrogen to supplement, because the moment you apply those 150 pounds, you inhibit nitrogen fixation. Why do you inhibit nitrogen fixation? Remember that I said that fixation is super expensive for the plant. The plant is pretty smart. If it has nitrogen in the soil, I'm not going to pay for those nodules. Too expensive. So you apply nitrogen, instead of fixing nitrogen from the air, you don't fix anything from the air. Those 150 pounds are not enough for high yields, and then you end up with lower yields at the end. Because remember, 4 pounds of nitrogen per bushel produced. If I apply 100 pounds of urea with 100% use efficiency, that gives me 10 or 100 pounds of urea, yeah, 10 bushels, 100 pounds of urea, if it's 100% use efficiency. Usually nitrogen use efficiency, remember, Penny talked about phosphorus use efficiency, nitrogen use efficiency is about 40, maybe 30, 45. So think 250 pounds of urea for 10 bushels, well, it doesn't pay that easy. It's not that easy. And if you apply it, you usually lose your nitrogen from the air. And we've done a ton of trials with that at the University of Illinois. This was a trial where, with no nitrogen, you see all the nodules. Here's where we applied urea banded 2 by 2, pretty much 0 nodules. Why? The plant is not going to establish the relationship if there is free nitrogen in the air. But we wanted to test it. And the idea we came up with is, why not supply nitrogen the closest we can to the moment those nodules stop working? Ideally, the moment those nodules stop working, if we have enough in the soil, well, maybe then we can keep up with plant demand and we don't remobilize as much nitrogen. And this idea came after we talked to Alex Harrell. He does this on his farm as well. All the acres, not only competition acres. He comes out at R3, the last time he can come in with Y drops before canopy closes, and he applies UAN Y drop. And we did that. So this was at R3, going to R4, the latest we could get in. We're looking at different rates of UAN, 40, 80, 120, slow release nitrogen, 40, 80, 120, or a combination of UAN plus slow release nitrogen. And these are the results from yield. What did we see? We did see that we were probably short on nitrogen. Slow release nitrogen didn't really work. I don't think it was released fast enough. We didn't get enough rain. UAN did work. Increasing rates increase the yield response. That tells me it is limited on nitrogen. Does it pay? Definitely not. It doesn't pay. But why is this so important for me? Because it's showing me that I do have a deficiency later on. It might not pay, but we gotta figure out how we can improve it otherwise. But this is telling me 5 bushels, and we applied this at R3. We spent almost a month without rain. So I feel like we lost a bunch by volatilization. We didn't incorporate as well as we wanted. So we're gonna repeat this trial this year, and I think the responses might be even bigger. But it's showing me we are probably deficient. Question?
Would like amino acids be placed in that position? Or could you?
The plant can absorb some amino acids through the roots, but it's minimal.
Or how about even like foliarly?
Oh, foliar?
Yeah.
It can. It can improve the metabolism, improve the plant overall health, and absolutely. But I don't think foliarly it's hard to get enough nitrogen in the plant. Amino acid is a form of nitrogen to make a difference. It's 4 pounds of nitrogen per bushel. How much do we apply foliarly? Maybe a pound or 2 of nitrogen, so half a bushel. You know what I mean? It's not that easy. We're gonna try this year with some foliar, because that's a question we got, especially the SRN through the leaf. That's one of the questions we got, and we want to see if we can do a combination of foliar plus soil and if that helps. Yeah, we found out that it's probably not good to apply our nitrogen. I wouldn't do that commercially yet, but we're seeing that we're probably deficient, and that's very important. So some scenarios where we do see a good response to nitrogen application, lower rates, 20 pounds of nitrogen, 100 pounds of AMS, ATS, whatever it is, providing 20, 25 pounds of nitrogen plus some sulfur, very good, especially early season. If you're planting early, your soils are cool, mineralization is not active, you need some nitrogen for that plant to pop up, have energy to establish that nodulation. A lot of times we see that plant not coming out of the soil healthy, and then it doesn't establish nodulation because it doesn't have enough energy. So lower rates of ATS, AMS can really help the plant early on, and that's a lot of the research that Sean Castillo, there's different researchers that looked at this sulfur with nitrogen early on, and it usually doesn't hurt nodulation because your soil is not mineralizing that much. Sometimes if you see going into the R stages, nodulation is not working, you can do some rescue nitrogen application or even a Y drop application of nitrogen. But I don't really think the soil is the key here. I really think improving the fertilizer or the nitrogen that we get from the air is key. And here I want to talk about one element that I've already touched on, but I think it's so important for nitrogen utilization, and that's why we see the responses we do see with it, and that's moly. We've been working with Molly, different ways of applying it on soybean quite a bit. So these are 13 environments, 2 application rates, looking at Molly applied to the seed on the planter box. Excellent results, excellent results. And we've done that back in, I think this is 2021, if I'm not mistaken, 22. We've been doing that for the last 5 years, and the responses are very, very good. And why is that? Again, remember the 2 enzymes I talked about? Nitrogenase has Molly, nitrate reductase has Molly. Remember the sulfur I talked about, AMS, ATS, apply some sulfur early on. You see these enzymes? They all have sulfur in it. We need sulfur for the nodulation, the nitrogen fixation to happen well as well. So don't forget your sulfur. Here's some published data looking at nitrogen uptake and nitrate reductase activity after a foliar molybdenum spray. This is 2 different years and this is 1 year only, but with Molly we're increasing total nitrogen uptake but also nitrate reductase enzyme activity. And a couple products that I want to talk about as a very efficient way of providing these micronutrients are these planter box products. You guys have offerings with planter box as well?
Yeah, so I was actually just looking it up. So the only thing we won't have in ours, and this is our lower end one, it has the same nutrient analysis. We don't have the rhodiola in there. But I mean, it's like $5 a pail. It's very, very high levels of Molly in it. So early on in the season, and the data is good on that.
Yeah, very good. So we have 2 products focused on soybeans, both with a higher concentration of Molly, 3% on Nutra, 10% on Homeland soybean. Here we also have an additional Brady rhizobium. And we've seen excellent results. This is a trial we had back in 2024. I collected the plants and extracted the nodules, and I separated by class or size categories.
Who did you piss off to get that job?
Yeah, that was my first month of work. I wanted to show some work, so
My God.
yeah, that was kind of annoying. But anyway,
Are you noticing a difference when you're having the enzymes, like from the PDS with the soybeans?
yeah,
Okay.
it's usually responsive. Yeah, absolutely. So here's untreated control. Here's a combination of ATS, PDS, and Homeland soybeans. We got 82% more total mass of nodules, meaning the weight of each individual nodules, but we also saw that we increased the bigger nodules. And why are bigger nodules so important? Because larger nodules have a higher ratio of nitrogen fixing tissue to overall nodule volume. So smaller nodules, most of it is just the skin or the peel around it. If you have a bigger one, you have much more of the inner content that is actually fixing nitrogen. So that's why bigger nodules means stronger, more efficient nodules. Here's some pictures, control compared to Homeland soybean. And here are the yield differences we saw that year. 106 compared to 101. Here's some other results. I only have a couple slides left. Here we're looking at the check. Talk plus microsurge. Microsurge is just Brady rhizobium. We usually don't see a response to Brady rhizobium in Illinois. What about you guys? I don't know if you guys usually inoculate here.
They do, but I mean, it's probably not as impactful.
Yeah. We usually don't see that much of a response because of the natural population we have in our soils. But when we add Molly, regardless of the way, and even zinc here in this case, very responsive, we see those big responses. And this was 2025. Untreated, Seedful Micro, this is our product geared towards corn. This is our soybean product, Homeland soybean and Enzyme Seedful Zinc. Not sure why Homeland soybean went down here. Maybe the Brady rhizobium caused some interaction. Not sure there. But yeah, that's what I had. I think I a little bit over an hour and a half, but talked a lot. I hope something was useful for you guys. I appreciate your attention and would love to answer any question if you have.
I mean, I look at, so last, I think 2 years we've been doing something similar to this, and what you've brought forward is kind of how the guy previous, it's a systems thing, it's not just one thing.
Yeah.
So if you really listen to what he was saying, I mean, he was focused on high yield, but I mean, we were talking about like basically 3 limiting factors there for the most part that you can do economically. Seed time, so on the seed, in furrow, post-herbicide spray time frame, stress mitigation, and then feeding late. Have you it doesn't, so you are seeing an economic gain if you're like going at your typical like R3, fungicide pass and fuller nutrition, then coming back again at like R6 with.
R5 is the data that we
R5.
have, yeah, but yes.
Okay.
Like what we usually say to growers, if you're not doing anything to your beans, because a lot of growers in Illinois aren't doing anything, apply fungicide. That's the first thing we would recommend.
Yep.
Apply fungicide at R3, that's gonna give you a couple bushels there, 3 bushels depending on the year. Very good. The second thing you can do, adding nutritional to that fungicide pass. BMO plus 3 or just BMO, born with moly. That moly is just so responsive, so responsive. Then a third thing, I would start to think about this stress mitigation, ascophyllum, but you don't have to do it all. But start adding some key ones that we really know at this stage, if you get in with this nutritional, it's usually going to give you a positive ROI. And soybeans are hard. Soybeans are not easy to understand.
How many here are happy with their soybean yields? I don't know a farmer that's ever said, yeah, I'm content.
You know, and guys, think about it, not that I want to break a price, but I mean, that BMO product has a lot of good data. That's half a bushel. That's what to make it pay.
Yeah.
It's less than a half a bushel. It's less than $5.
So in comparison to other things or products coming to like on the streamlined Brandt side, you look at all that stuff and you're thinking in your head like, oh man, what's this gonna cost me 5 bushel? I mean, what is?
I mean, I just
It's
did the
not
math. You can do
bushel?
Surge S7 or Seaweed Max S17 and BMO for $12.5. Okay, so you need roughly like 1.1 bushel to make that pay. I would argue from what we saw last year. Now, last year was pretty extreme because of how we finished out the year. So when we were seeing 5, 6, 7 bushel responses, I mean, geez, I feel pretty confident. I mean, the data's consistently showing 2.5, 3 bushels.
That's the first thing I noticed when Blake and I sat down and going through all this stuff. I was like, all right, this is gonna cost the guy $30 an acre and not even close.
Yeah. We know that if the product is too expensive, it just won't flow through retail, it won't flow through distribution. So we want the product getting to the grower at a bushel of corn, $4, $5, $6 maximum. So, and usually has to have like a 2.5 ROI. Like we want it to be getting the guy 2 to 3 bushels of beans. So beans are not easy, but if you plant early, good singulation, pay attention to this early development. If it's you guys have very high organic matter soils. Your climate is even colder than we are in Illinois. So until this soil starts get activated, you need some nutritional. So maybe that 100 pounds of AMS, ATS, or a little bit of enzymes to activate that soil is super important.
So you were talking back there, and this wasn't part of your presentation, but you were talking about zinc on soybeans.
Yeah.
We use ends up zinc, or zinc up is what we call it, on corn. Would that be a good option to run a quart of that in fertile if you got the availability to do that? Especially here, you know, we're planting soybeans earlier and earlier. Those enzymes are activating that soil.
Absolutely.
What do you think of that? Like,
Zinc is so important for. I showed you guys about the stress.
Yep.
So these plants are planting under cool temperatures. A lot of stress is going on.
Yep.
It's utilizing only what it has in the reserves. So adding some zinc to it really activates this antioxidant system. But we're seeing faster, more uniform emergence with the zinc applied to the seed, and fertile would work as well. And the enzymes activating the soil. And the analogy I always like to make is think about why do you put food in the fridge? So it doesn't go bad, so it doesn't spoil, right? What is the fridge doing? It's lowering the rate of microbe reproduction, bacteria and fungi. So if your soil is at the temperature of a fridge, there's no microbial activity.
Never heard that analogy before.
And if you don't have the microbes active in your soil, all that nutrition that you have in this beautiful black soil is not available, unfortunately. Nitrogen, sulfur, phosphorus, very important nutrients. So a lot of times we're planting these beans super early, but it just takes so long for them to take off because it's cool, it doesn't have enough nutrients available. So getting them started is super important as well. And then utilizing an enzyme which really activates the soil, releases nutrition, zinc and some sulfur can be really, really good.
and one thing, and maybe Marcos can speak on this, I mean, I used to work for a different company. I'm not against microbes at all. It just seems like the consistency, you know exactly what you're getting with these enzymes. You know the exact count you're getting in. We know that they're in the soil for up to 6 weeks, basically, and they're very, very specific on what they are doing. And Brandt, frankly, has the data to back it. I mean, I was going back in all the research books. They got data from Zinc Up from like, what, 2012?
No, probably not, but
I mean,
we've been
long
working
ways
with
away.
them for a long time.
Yeah.
But I can't forget to tell you guys, we spent a lot of time writing our research book every year, and ours just came out last week. Unfortunately, I don't have the printed version here with me because we didn't get it printed, but it's on Brandt's website. This year is almost 90 pages with all the research we've done at the research farm, but also third party, and there's a lot of educational sections explaining what we're doing and why we're doing. So it's a pretty neat book that Eric and I, we wrote most of it, so there's a lot of information in there. But on your point there, Blake, actually I was listening to this interview with some key guys in the biological industry, and they say that the future is probably, there will be some living microbes, but most of the future in the biostimulant biological categories are going to be with the metabolites from these biologicals. Because the moment you go out, like I see you guys sell pivot, for example, the microbe doesn't do anything. The enzymes that microbe produces does the excrete, exactly. So instead of applying the living microbe and expect it to reproduce, survive in the soil, and then excrete those substances, what we're doing is have them produce it in the lab, in the facility. We concentrate them and we apply the actual enzymes or the metabolites, which is
trying to skip a step.
Yeah, shelf life is much higher, years of shelf life. It doesn't matter the temperature that much. You don't have to worry about it being active or not. The moment it gets in the soil, it's active.
And I would argue that we're probably going to see, from what I've observed, we will probably see a little bit higher responses with running those enzymes in fertile, because we are going to be colder than where that Pleasant Plains location's at. I mean, heck, you guys were planting beans March 15th, weren't you, last year?
Yeah.
Your first trials.
Yeah,
We might be this year.
we still lost 60% of our stand, but it's
Yeah, but still, we don't have the opportunity really to do that. So I mean, we are dealing with cooler soils, and if we want to get, he showed you, the best thing you can do for soybeans is earlier planting. You saw better responses, okay? That's a good way to get those enzymes working. We know exactly what we're getting. I know you had a question.
So you talked about foliar feeding and towards the end of the reproductive stage or whatever. And I think about like what the weather's like that time of year. Like it's hot. Do these products burn the plant, hurt the plant? Is there a better time to apply them?
Oh, definitely.
Or, I mean, does it make a difference?
It does, absolutely. If you're gonna spray, it's 110 out. Better at the end of the day or early in the morning. It's absolutely gonna be better because when it's so hot, the plant stops. It closes the stomach. It won't absorb and move. So it's better. Of course, our product stays wet for longer. So at night, it's gonna start absorbing. But I would rather do it either very early in the morning or later in the day, so it has a night to actually process all of that, and it does make a difference, absolutely. But these products won't necessarily burn. Their composition, we have like some sugar alcohols that are actually good. You know when you buy like a soap or like stuff to hydrate your hand, it says like glycerol? Glycerol is a sugar alcohol. They add it to cookies, they add it to cakes to keep it moist for a long time in the shelf. What is it doing? Attracting a lot of water. We have that in our product. So it attracts a lot of water, it keeps the water, that molecule in solution, it absorbs for longer, it doesn't stress, it's not as strong. So, but absolutely, time makes a difference.
So you're saying you're keeping the water wet?
We're keeping, yeah, we're avoiding the water from going away. Put it that way. Yeah, appreciate it, guys. Thank you.
You talked tonight